Hello, uh, hello and welcome to Biomentors. This is the second video lecture from the series Developmental Biology, in which we are going to discuss about basic terminologies in development. And in this video, we are going to mainly focus upon stem cells and its type, as well as we are going to see induction and competence. What is that? And we will also study it by an example. So let's get started. So first of all, what are stem cells? So stem cells are a type of cells which uh, which uh, which has the activity to divide, and the activity of division is such that they have self renewal property. They are undifferentiated cells, which means they are not differentiated, so they can form all cell types, right? So basically, there are three properties of stem cells from the definition. Like, it can divide, it has self-renewal property, and they are undifferentiated mass of cells. Different types of stem cell division is seen, in which there are two types of division: symmetrical division and asymmetrical division. In symmetrical division, um, a stem cells, when it divides, it produces two type of cells, and they are of same type, which means either the stem cell will form two type of stem cells, or they will be forming two committed cells. So this phenomena is very rarely seen, and it results in expansion or reduction of resident stem cells. So it balances the number of stem cells in an environment. Okay, that is homeostatic. And the second division is called as asymmetrical division. This is the normal cell division of any stem cells at normal conditions, and they are mostly seen. This is also called as single cell stem cells asymmetry, and in this case, a normal stem cell division takes place in which one stem cell is formed and second committed cell is formed, as seen in the figure. And this stem cell stabilizes stem cell population itself. All right. So these are the two divisions of stem cells. Now there are two types of stem cells. First one is embryonic stem cell, and second one is called as adult stem cells. Embryonic stem cells are pluripotent in nature. All right. So they are true stem cells, and adult stem cells are tissue specific. They can be multipotent or unipotent, and they mainly repair the tissues. Okay, they mainly repair the tissues of any organ or any body part. So now, there is very wide stem cell research going on at large scale. So induced pluripotent stem cells was discovered by was discovered, and it is called as Yamanaka factors. So what are these Yamanaka factors? Basically, they are transcriptional factors, and there are four types of major Yamanaka factors, that are SOX2, OCT4, CMYC, and KLF4. So SOX2 and OCT4 mainly activates Legon, which establishes pluripotency and blocks the differentiation of cells. And secondly, CMYC opens the chromosome, which should be accessible to OCT and SOX2. And KLF4 prevents the cell death. So these are the functions of different transcriptional factors, which has potential which has potential of pluripotency. So from this we can generate a unipotent cell to a pluripotent cell. All right. So this now we will be looking upon what is induction and competence. So induction and competence is a process in which it is basically a signaling process, in which inductions, which means induction is caused by inducer cells. So inducer cells mainly gives signal, and competence means ability to respond to a specific signals. So they have ability to respond to a certain type of signals, and thus as it is responding to certain type of signals, such cells which have competence are called as responder cells. All right. So first of all, as seen in this figure, inducer cells inductive signals uh, will send inductive signals to the responder cells, and then after responder cells receives this signal, and then after certain downstream processing would be done. All right. And particular transcriptional factors uh, or any other thing would be happening. So induction and competence mainly deals with two types of signaling, which is paracrine, or it might deal with juxtacrine signaling. So we will be looking upon Island's induction in amphibians. So in amphibians, there are two types of cells. One act as an inducer and the second act as a responder. So in inducer is acted by optic vesicles, and head ectoderm are the responding cells. So optic vesicles have certain type of signaling molecules. These signaling molecules are BMP4 and FGF8, and the head ectoderm has receptors and different pathway run inside these cells. So head ectoderm has a special type of receptor TGF beta for BMP4 signaling molecules, and RTK receptors are there for FGF8, and different type of transcription factors SOX2, which is generated by TGF beta family, and LMF by RTK pathway is activated. This both are the transcriptional factor, and one competent factor is also present, which is present in the responder cell itself. So PAX6 is the competence factor, and mind well, it is not a induce it does not come from inducer cells. It is already present in the responding cells. So in this figure we are seeing that BMP4 and FGF8 both are released by the optic vesicles. In head ectoderm has different receptors, namely TGF beta for BMP4 and RTK for FGF8 is there, and they receives that signaling molecules, and a pathway runs inside the head ectoderm cells or the responding cells. So TGF beta binds to this TGF BMP4 binds to the TGF beta receptors, and SMAD proteins are activated. This SMAD proteins, namely SMAD1 and SMAD4, together with the SMAD5 proteins, SOX2 is activated, and in the RTK receptor pathway, RAS and RAF are activated, and then finally MYC and MAC kinase are activated, which activates transcriptional factor LMF. All the two transcriptional factor and the competent factor, namely SOX2 and LMF, and PAX, which is a competent factor. All these three factors activates delta crystalline genes, and then after lens induction is seen in amphibians. So this was the basic concept of induction and competence, and there are two modes of inductive interactions, which means there are two types of inductive interactions. First is instructive interactions, and second is permissive interactions. In instructive interactions, responding cells has a two fate. If it is induced by signal, then it will show a different fate, and if it does not get inductive signals, then it will show their default fate. The second is permissive interactions. The cells are already committed to a certain type of fate, but the cells always require an inductive signal to perform that type of to achieve that type of way. This both are different, and through various examples we would be studying this type of interactions. This was all about stem cells and induction and competence. In the next video we will see what is cleavage and what are different type of cleavage. So if you like this video, please do subscribe and hit the like button.